Okay, so one of the issues that we need to think about anytime we are conducting psychological research is how we can avoid bias in our research. And bias is a pretty nebulous term. There's actually a lot of very specific types that we need to keep an eye out for so that we can avoid them. And keep in mind that these apply to all different research methods, surveys, experiments, whatever. So the first one we have to look out for is sampling bias. And this is what happens when you have a non-representative sample. So remember that there's always a group of people that's your population that you want to answer questions about, and your sample needs to look like your population. If you want to know about all Americans, but you only survey men or you only survey teenagers, you're going to have a biased sample. Second one we have to look out for is called volunteer bias. The people that are willing to be in your study might not represent the entire population. Now, this is a much bigger issue if it's a study that's going to take a lot of time or be on a sensitive topic. So, if you want to do a survey and ask people really in-depth questions about their sex lives, well, the people that are willing to tell a stranger about their sex lives, maybe they're different from the people that just say, no way, I'm not going to be in your study. And so now you may have a subset of people who don't represent the population as a whole. Third thing we have to watch out for is experimenter expectancy effects. It's sometimes called experimenter bias or researcher bias. And this is not about fraud, which is totally different, but this is about when you have researchers who just are human. And so their bias might affect how they collect data or how they interpret it. If I am a researcher and I interview someone about how much they like psychology, right? Well, I love psychology. So without realizing it, I might say things like, yeah, you find psychology fascinating, don't you? And I'm nodding my head. And so this is kind of causing them to change their behavior because my bias affected my behavior. Now, in that example, if that person changes their behavior based on what they expected, based on what they thought I expected, that would be called demand characteristics. So that's where the participant changes their behavior based on what they think the researcher is looking for. So now, because I'm nodding my head and asking the question a certain way, maybe they change their answer and I nod my head more and they make their answer even more extreme. And so we can kind of go back and forth where my bias affects my behavior and it pulls for certain behaviors from my participants. And then the last thing we have to watch out for is what's called placebo effects and nocebo effects. So placebo effect is when a person improves based solely on expectations. So like somebody takes a pill and they got better just because they took a pill, not because of the pill's active ingredients. So that's the placebo effect. Nocebo is just the opposite. People experience harm because they expect to experience harm. And usually when people think of placebos, they think of sugar pills, but you could also have placebo acupuncture, placebo injections, placebo surgery. Anything you do, if it makes someone get better due to expectations, then it was a placebo. And so when we want to control for all these different kinds of biases, sometimes, and the key word here is sometimes, we can get rid of these biases by keeping the participants or the researchers blind. And blind means they don't know who's in which group. So if it's a drug study, for example, everybody's going to be blind. The participants don't know which pill they get, the researcher doesn't know which pill they get, and after they collect all the data, then they go back to the list to figure out who got which pill. So that's a double-blind study. But sometimes you can't keep everybody blind. So for example, I once did an experiment where some of my students had take-home quizzes and some didn't. Well, I can't keep my students blind. They're going to know if they took a quiz, right? I can't prevent them from knowing that. But I could still grade their papers blindly by putting an ID number instead of a name on the paper so that I grade it without knowing which group a student was in. And then after I assign the grades, I say, oh, student 25 got a B. Now I go back and figure out who student 25 was. So this is a way that you can control sometimes for experimenter expectancy effects, for demand characteristics, and for placebo effects, but not always. And keeping people blind will not help with either sampling bias or volunteer bias. But this whole idea of keeping people blind, this doesn't actually get rid of placebo effects. What it does is it gives us a baseline. So if I have one group that's taking a real drug and one group that's taking a placebo drug, everybody is going to get better to a certain degree due to placebo effects. What I want to know is, did the drug group get better even more? So I'm not saying did the drug work better than nothing. I'm asking if it worked better than a placebo effect. And that's it for bias. If you have any questions, please feel free to contact me. Otherwise, I hope you all have a great day.